Since this is my third lecture, I thought I'd wrap up some of the things that I started in my first two lectures, giving you a little bit of an overview about violations which prevent you from doing some of the more interesting things that I presented in my first two lectures. But the violations are actually interesting in their own right and really kind of cool to think about in the context of coalescent theory, as well as in terms of how do you go about identifying these violations. So before I do that, let's just talk about what I think we've learned, hopefully all of us, in the two lectures that I've given and in the two lectures that Lucy has given already, which is that protein sequence function has this somewhat ambiguous but getting more and more clear kind of timeline. And if you think about amino acid as a string sequence that contains information, of course, one amino acid sequence contains enough information for the cell to produce a protein. But for us to be able to infer what the specific importance of each amino acid position is, it of course helps to have several versions of that amino acid string, right? So variants from one species, second species, third species. And I'd like you to sort of think about 4 pieces of information that are possible to take away from a really good, dense, multiple sequence alignment. This does not have to be exclusively at the protein sequence. This really could be a string of any characters, right? So the phylogeny is agnostic to what is exactly being measured here. There's some sort of matrix of transition between different states. This could be nucleotide, this could be protein. At the RNA level, it could also represent RNA sequences. It could be digits on the hands of cartoon characters. Doesn't matter, right? Transition states. What are the things that we think about or should pay attention to? Well, if you have a dense set of multiple alignment, the first thing that you think about and that your eye is drawn to are positions that are completely conserved no matter what the divergence level, right? So if you're, let's say you're comparing isocitrate dehydrogenase from all kingdoms of life, what you will find, if you have done a decent job with your multiple alignment, is that the catalytic residues of isocitrate dehydrogenase are absolutely universally the same. It doesn't matter whether this is bacteria or archaea or eukaryotes, right? So that's the very, very strict conservation that tells you that natural selection via mutation must have been afforded lots of opportunities to move away from that sequence, and it has not. So that's very, very strict conservation. I'm going to lump into that category semi-strict conservation, where you tolerate a couple of biochemically similar residues, but there's clearly constraint. Whether that's constraint is at the level of catalysis or is the level of protein folding, we don't know based on just primary string of sequences, but you can infer constraint quite nicely based on that. What I talked about in my first two lectures is sort of the opposite end of the spectrum, where that position is so rapidly evolving that every character in this multiple alignment appears to be distinct. And traditionally, molecular biologists have actually ruled those out or not paid much attention to those, because after all, if they were important, evolution would have constrained them. And now we know that actually, if you think about them in the context of red queen dynamics, those could be very important specificity determinants, right? So we've now gone from very constrained equals important to potentially very rapidly evolving could also equal important. Now, I want to be very careful here that that does not mean that all lack of constrained positions are important. In fact, most lack of constrained positions are highly unimportant. It's important that there's an amino acid there, but the nature of that amino acid doesn't matter. So these are things where you have loops of proteins where you can maybe tolerate 18 out of the 20 amino acids. Proline is prohibited pretty much everywhere. And so that's pretty unconstrained. That's not positively selected. That's just sort of neutrally evolving at the cusp of we need something there, but what that something is perhaps not so important, right? So that's sort of the first level of, very constrained, not at all constrained, to very rapidly evolving. I'd sort of like you to think about those as 3 distinct categories. For a typical amino acid that has some sort of preservation across, let's say, all animals, we tend to find a lot in the very constrained positions when you think about protein sequences. We find very few instances of very rapidly evolving. This is positive selection is a rare phenomenon. I just want to emphasize that. It's both rare in the number of genes that are being affected, but also in the number of codons in a gene. PKR is unusual, MXA is more usual. Small proportion of genes of amino acid residues affected. But there are, we are encountering more and more things like disordered domains, proteins involved in phase transitions, etc. And they're somewhat violating our assumptions based on this very simple sort of 3 separation thing. But what we are finding, again, through deep mutational scanning and other mutational analysis is, in fact, there are many positions that are conserved, but when you test them in the laboratory, we don't know why, because they do, in fact, tolerate a lot of mutations. That reflects 2 things. Our ability to fully see sort of what is the fitness landscape of a protein is somewhat limited to the assay we can do, and perhaps we are not fully capturing all the interaction space of that protein with other binding partner proteins, and that's actually going to be a focus of work in the coming years. Lucy also talked, and she's actually an expert about thinking about covariation or mutual sort of covariation across positions. Of course, if you have completely conserved residues, there's just no possibility of covariation, right? So you need at least some sort of change in order to do that. Turns out that if you're very rapidly evolving, that signal also abrades really quickly. So again, in that sweet spot between very conserved to very rapidly evolving, you have now the potential to think about what has been referred to as protein sectors, et cetera, pieces of protein that are interacting with each other, perhaps because of allostery or other interactions. So at every step of this conservation landscape, there's information to be gleaned out. And it's kind of interesting to be thinking about these because these strings of characters, as I said, can be basically treated as mathematical strings of characters and give you inferences, yeah? Assumption to all of this is we are making an apples to apples comparison, right? Which means that there is an underlying phylogenetic tree. It accurately reflects what is happening for most of the sequence, the string of characters. No matter which string we take, they can be represented by the same tree. I'm now going to present some cases where that is not true, and because of that violation, you actually have, if you were to sort of ignore blissfully what has been going on in terms of potential recombination or horizontal gene transfer, you may miss some very interesting biology, all right? So everybody knows what an ortholog is? Okay, maybe I should switch that around. What's an ortholog? Okay, an ortholog means that this is a string of gene sequence, perhaps a protein coding gene, that is completely related by speciation, right? So if the species diverged, those gene sequences diverged exactly at the same spot. Whereas a paralog is are 2 gene sequences that are related by gene duplication. They're not at all related to when the sort of species or the genomes carrying them separated. That's sort of a simple poor man's definition, but it's good enough for us right now, right? Which means that you can have 2 sequences that are related either by speciation or they're related because of gene duplication. And those can be treated very differently. When you have paralogs, you can actually have recombination between them, which then increases the possibility that you've got skewed phylogenetic histories, which can actually skew all of the inferences that I told you about. And we'll just look at a couple of case studies today. Okay, everybody on the same page? Okay, so I'm first going to tell you about this phenomenon, which is rampant in viral genomes, called overprinting. Does anybody know what overprinting is? Makes you think about what printing or underprinting is, right? Like, what's overprinting? Actually, overprinting is a really cool phenomenon, and it arises because of our implicit assumption that when we have a protein coding DNA, and you produce a string of amino acids associated with that, we always tend to think about it occurring in one frame, right? So CCT triplet codes for a proline, ATT codes for an isoleucine, and so on and so forth. But there are many instances where this protein coding gene can be coding for 2 completely unrelated strings of amino acids, because in the frame off by 1, you have CTA, which encodes for a leucine, TTA that codes for a tyrosine, etc., right? So why do you see this? Well, you sometimes see this because viral genomes are really limited in their genome size, and to maximize the complexity that they can actually encode, given the constraints of the small genome size, they basically use the same piece of DNA to encode multiple open reading frames, right? So these are all protein coding genes, but they're basically twice the protein coding gene per base pair, if you will, than a normal case. So how do we go about identifying these? Well, there's multiple metrics, but using the intuition that you've already learned, how would you go about looking for case studies of overprinting? I should have said, how would you means question, expecting response. So there's no bad answers, but so you have every piece of information you need to come up with a guess, like how would you go about assuming that you have, multiple versions of this DNA sequence? How do you know is this encoding for one protein coding frame or multiple? Yeah. Would you be able to, if this was a completely hypothetical second frame, you would probably still get some position weight matrix, right? Because the second codon would become the first codon, and that's a constraint that would basically be translating. You're very close. I just want you to be thinking about even simpler. Yeah.
3L, you did the deep sequencing.
Yeah.
So, and you got to know that with the help of, like, in case of, with reference to the reference genome, there was more of K3L in replication. Mm-hmm. So if you do the deep sequencing here, will you see some like peak in this thing?
Maybe, but it's a different kind of peak. I should emphasize, deep sequencing is bloody expensive. So your first instinct should not be thinking deep sequencing. It should be like, what's the cheapest experiment that I can do? There is a much, much cheaper version. Go ahead. Exactly. Why? Because what is a synonymous codon in this case? What is a synonymous codon, which means a silent site mutation like this position here, is now a replacement change in the other frame, right? So now this piece of DNA has way fewer actual silent site mutations because, some frame basically considers that an amino acid replacement, right? So that means that DNA sequence is really locked in because of the fact that it's basically forced to preserve the protein coding capacity. So let's sort of take a look at that. So here's the HIV genome, and what you basically highlighted in multiple instances of completely distinct, completely unrelated to the amino acid sequence, protein coding genes that are basically encoded by overprinting from different parts of the same base genome, right? And if you look at this part of the genome, what you basically find is the net DNA sequence mutation rate is far lower, right? Because you're actually preserving both of the open reading frames that are basically coming off this piece of DNA. Is that clear? Everybody on the same page? Okay. So, if there was a synonymous change here, let's say at ATA, you'll realize that actually becomes a stop codon in the other frame, TA. So that would be obviously very bad if your intention is to simultaneously encode two different protein coding genes. So that doesn't mean you cannot have changes. That just means that the spectrum of mutation is far lower. Now, it might surprise you that this is just not a trick that viruses use. In fact, there are many human genes that are overprinted in different parts. There are some important pairs of oncogenes that have overlapping frames going in the opposite direction, where you're basically encoding one amino acid string in one direction and the end of another protein coding gene in the other direction. So lots of constraint happening, lots of sort of hypermutation spots associated with those kinds of changes. So we should observe a depression of synonymous mutations, specifically in the overprinted part of the gene, right? So I'll just give you an example of how we went about this. There's many, many studies. This is just one that I picked because I happen to have the slides, because this was work that we did in the lab. And this came from Merkel cell polyomavirus. Polyomaviruses are very small DNA viruses. There are multiple distinct human viruses. This particular one was isolated as the causal agent of a deadly skin cancer disease called Merkel cell. I mean, that's the survival rate with Merkel cell is very low. And we know the etiology of this disease. It comes because of insertion of this polyomavirus. Anybody else of any other, well, I guess they're on this page, but any other human polyomaviruses that have been in the news? Okay. You don't read your emerging infectious diseases as closely as I do, but there's been a couple identified also as potential agents of other types of skin cancer. SV40, which if you're a biologist, you at some point use the promoter from SV40 in human cells because that's like the promoter that really is the gangbuster promoter, but it actually comes from a virus. There is a mouse version of polyomavirus called mouse polyomavirus, which causes cancer in rodents, and of course, Merkel cell that causes cancer in humans. So this story starts off with a perplexing set of observations that come by comparison of different polyomaviral genomes, including this JC polyomavirus that has this large T antigen gene. Now, the large T antigen is the gene that basically hijacks all of the cell cycle control in the human cell through different domains. It takes over P53, it takes over retinoblastoma. It is basically the protein that the virus is using to completely convert the human cell into a polyoma production factory, right? Super important is the largest gene encoded by the polyoma viruses. And these guys have these characteristic domains associated with these functions. So there's this DNA J, which is a sort of a DNA binding domain, ATPase helicase domain, this OBD, which is another DNA binding domain, and these two domains that interact specific with cell cycle proteins. But what I'm sort of using this to illustrate is the fact that these large T antigens are very variable in size. So this one is 688. This is not even the smallest. There's one that's only 500. And then the Merkel cell, which is like 817. So people have actually speculated that this is basically some sort of junk, which is what is accumulated in Merkel cell, which is sort of one of those things that you say because you don't have a good argument why that would be. But because this is a virus, you sort of don't believe it yourself, even though you're saying it, because there should be a lot of constraint on the size. So what we decided to do was actually do a sort of slightly more sophisticated analysis, which is just look at DS. Remember, we're not paying attention to DN. We don't care about the amino acid coding capacity of preservation. We're simply looking at synonymous sites. Now, if you buy the hypothesis that synonymous sites are mostly neutral, they should accumulate as a sort of rough proxy of mutation rates, they should be equally distributed over the polyomaviral genome and over the large T antigen. But you can see here that there is in fact this region of depression, even this crude sliding window analysis, exactly coinciding with the potential to encode another open reading frame of roughly 300 amino acids. And we call this ALTO for alternate T antigen open reading frame. And this is now in the +1 frame, so it's off by 1 nucleotide. It makes 248 amino acids, sorry. There's really no recognizable homology here, right? So here, if you had to gone about relying on position weight matrices, it would be a tough ask, right? So at this point, you should be skeptical. Well, there's this depression in DS. There's a potential for an open reading frame, but because there's no similarity to anything else, just a sort of hydrophobic C-terminus, which we know now is used to anchor it to the membrane, we have no idea what's going on. But there is a potential that this is in fact a completely new open reading frame in a cancer-causing polyomavirus. I know lunch was really great, but please do feel free to stop and ask any questions, yeah? Okay. So we said, well, okay, we don't have any protein homology, but perhaps we can look for the same preservation of the open reading frame in other related polyomaviruses. So here's Merkel cell, and here's the closest polyomaviruses. And so here's the stop codon in the ALTO frame. So here's the ALTO open reading frame. And what immediately caught our attention was the fact that there was, in fact, a stop codon almost precisely at that position in a whole bunch of related polyomaviruses, right? That could be by chance, but the more you actually accumulate, the less chancy it looks like it's becoming. Like, what's the likelihood that you, within 4 or 5 amino acids, have this? And then you can see many of these had the potential to encode an open reading frame in the ALTO-like frame. Whereas after a certain point in evolution, you had this stop codon, but you had stop codons everywhere. So no potential to encode an alternate reading frame, which meant that if we were right, and ALTO was a bona fide open reading frame, this was not actually present in all polyomaviruses. It was actually present in a subset of polyomaviruses that includes Merkel cell polyomavirus. Okay? So here's a phylogeny based on other genes of the polyomaviruses, and at some point here, you basically gained the ability to encode this alternate open reading frame from the large T antigen. Another way of showing that, yes. How far back do you want me to go? Here? Well, so we would say that this is distinctly non-neutral because we have a depression in the DS, which should be consistent with the fact that what is DS, or synonymous sites in the large T antigen frame, is actually a replacement site in the ALTO frame. Sorry, what? This is not similar to hitchhiking signal, no. Yeah, it's a good question though, because what we basically have on top of that is the complete co-occurrence of the preservation of an open reading frame. So this could be hitchhiking signal because you basically have an open reading frame, but we are basically averaging over multiple completely distinct polyomaviruses here, right? This is a sliding window of, these are not variants or strains of the same polyomavirus, they're completely distinct polyomaviruses, like a pairwise comparison. These are 3 sequences that we use to, do like a sanity check, but we can extend this quite a bit farther. Okay, so at some point in polyomaviruses, over here, these guys all have the potential to encode this sort of alternate open reading frame. By the way, at this point, I haven't shown you any evidence that this frame exists, right? I'm just building the case in a slow way. You should be just as skeptical as we are at this point. Yes. In the large T antigen, in the one protein that we know that is actually encoded. We are not making a distinction between biochemically similar things, because once again, we would not expect a depression in just one part of the gene there, right? We are basically averaging DS across the entire genome, and even in the large T antigen, we have DS being about the same as it is in the rest of the genome, but it's really low in one part of the genome. So think about it this way. There could be many reasons why you have a depression in DS. You could have an RNA element there, right? So there's some secondary structure of RNA that's super important. You can't mess with that. So DS is simply telling you that your fundamental null hypothesis of there being one open reading frame and nothing else is false. It doesn't tell you that there's a second open reading frame here. That's what all this downstream work is there. It could be a promoter or an enhancer or some other element that is under preservation, which is distinct from the preservation of a protein coding gene in the original frame. Got it? Yeah. Okay, so I'm going to actually tell you what we think is going on and then show you the data. Actually, turns out we became interested in understanding, well, how does this happen? How do you go from a viral lineage which just spontaneously seems to birth an entirely new gene in this alternate open reading frame? I mean, this seems like a fantastical kind of idea. I should point out, though, this is actually getting less and less fantastical because we know now of at least 55 genes in the human genome that encode protein coding open reading frames because we have evidence from proteomic data. And we know for a fact that in chimpanzees, gorillas, orangutan, they're actually part of untranslated regions. So what was previously a completely non-protein coding gene, just by the fact that it does not encode a stop codon and has acquired a start codon, gains the potential of becoming sort of a bona fide de novo gene. And 2 of those are completely important for human male fertility. So this is like a something from nothing phenomenon that traditionally molecular evolutionary biologists have completely ignored because they felt like it's like chimpanzees typing on a typewriter. No matter what they do, they're not going to come up with Shakespeare. It's not Shakespeare, but it's at least a limerick. And apparently the limerick is really important, right? So this is kind of like what we think is going on. But what actually helped ALTO come into frame was the fact that because of the conservation of residues in the original frame, we were also basically creating constraints for the start codon and the hydrophobic motif of the ALTO frame. And I'll show you evidence for that in a second. But there were a whole bunch of stop codons separating that start and the stop codon. What we basically then had was an expansion of the large T antigen, potentially expanding the open reading frame in the ALTO frame. Again, this is all neutral. There's no selection because ALTO has not come into existence yet. But as soon as those stop codons moved, and we think that this was completely stochastic, boom, you suddenly discover you have this completely novel protein coding capacity. It happens to be important because it helps control the host cell cycle in a different way. And now that's complete. Once it's come into existence, it's completely preserved in both mouse polyomavirus and human and all the viruses related to that. So this is a something from nothing phenomenon. This totally violates your assumption of comparing orthologs across many different lineages because this doesn't even exist, in every sense of the word in many related polyomaviruses. Does that make sense? You guys with me? I just want to get a sense of like what the level of skepticism here is like. Is it 100%, 90%? No, it's not. This was, MT stands for the middle T antigen gene, which is what it's actually called in mouse polyomavirus. So it's a more of a historical thing than nothing else. Okay. So what's kind of interesting, just since you've asked the question, is we knew that there was in fact an ALTO-like gene, no homology by the way, except for the hydrophobic motif, which we thought was generated by alternate splicing, but people had amazingly missed the fact that it's an overprinted gene, despite the fact that the protein had been discovered. Okay, so what was in place way before ALTO came into existence? So let's take a look at what are the features you need to make a protein coding gene. You need a start codon. You need a stop codon, and you need no stop codons between those, right? Like that's the three features you need. So let's look at the area corresponding to the ALTO start codon. So I told you about this motif that is absolutely essential for large T antigen function, right? So you see this motif here, YGST, right? So what is that? That is a sequence that encodes for the completely essential YG motif involved for RB interaction. Great, but just one nucleotide over, you basically end up with this, which is the methionine and the valine, which is in fact the completely conserved stop. So actually, you didn't even have to invent a start codon in the alternate frame. It was preserved, presented for you long before ALTO came into existence by virtue of the preservation in the alternate frame, the parental sort of ancestral open reading frame. Is that clear? Okay. So that's, by the way, true. There are representatives here which have ALTO, but there are also representatives here that don't have ALTO, but they still have that methionine. So that proves that the methionine actually pre-existed even before the ALTO frame came into being. Okay, second point, let's look at the other end of the spectrum. Here we have this OBD domain, and the OBD domain is really important, but actually in one frame over, that leads to all of these hydrophobic residues basically being preserved in the ALTO frame. So again, the hydrophobic motif, which we now know through downstream work is important for ALTO function, was not invented by ALTO. It just came into existence already because of the preservation of the ancestral open reading frame. Why do we know that? Because if you look at the guys that don't encode ALTO, they're also hydrophobic, except they happen to be smattered with stop codons. But the hydrophobic nature of that open reading frame is already pre-existing, right? I just want to, if you are skeptical still, it's totally okay, because this was actually a big surprise to us, the fact that the start and the stop codons were in the context that was already present. We have some sign language going on here. What's going on? Well, we know that this is a hydrophobic patch that is long enough to integrate into the membrane. And so then we get excited because that's an anchor residue that can cross a lipid bilayer, right? It needs to be 15 amino acids long. It needs to be hydrophobic of character. And that's basically a membrane anchoring domain, very common. Low bioinformatic content because it tolerates a lot of change. The nature of the residue is not important as long as they're hydrophobic. Is that what you're asking? Suresh, you have a question? I don't think there's anything that prevents that from happening. It just happens that this thing was one. Some of the de novo genes that I talked to you about, I'm sorry, some of the overprinted genes in the human genome are actually in the -3 direction, like -2 direction. So basically, minus means you're going in the opposite direction, right? So there's not even that consistent frame there. It just happens to be you arrive at that stochastically, completely neutrally. It's kind of like, I don't know how many of you were at this paleovirology lecture that I came. The retrovirus came in, it brought an envelope gene. There was, life was perfectly fine before the retrovirus came, but once the envelope gene came, it's like, well, I'm going to use this. This is kind of like what happened here, right? Evolution takes whatever it has at its disposal. Okay. I think it's a little bit more, is a little bit less complicated than what you're suggesting. What I would say is that the large T antigen was always an open reading frame, and it has always remained an open reading frame because it's very important. But in the context of that one open reading frame, you can invent de novo a completely new open reading frame just being in a different frame. Does that make sense? I mean, so let's think hypothetically. This is not what happened in ALTO. Let's assume that you have this large T antigen, and you suddenly acquired the ability to initiate translation here, right? And you're now making both a long T antigen and a short T antigen. But if the short T antigen turned out to be something that evolution liked, you basically preserve those. Those are now both features that you would preserve, right? You acquire those features stochastically, but you preserve them by natural selection. Yeah? Are you with me? I don't know. I don't know whether that's like I agree with you or like I don't know what you're talking about. Okay, fine. Let's assume you're agreeing with me. It's totally an accident that it has proper function, but I think that's a little bit of an overstatement. I think it's had some function, but once it existed, natural selection could actually act on to refine it. And that happened early on, right? So we don't have a means to reconstruct how it was born. And I'm going to show you some cases where you actually have compromised the large T antigen to preserve ALTO. Yeah.
So talking about hydrophobic, right? You said that it's hydrophobic, right? So somehow the fact that the protein that you're coding for already has that function, you also
yeah, but it's actually a little bit, again, more complicated than that. I'd love to say that that's true, but if you notice, these are the guys that don't encode ALTO. You know, all the non-stop codons fit the pattern, but there's also stop codons there. So this is not like a preserved hydrophobic patch. It just happened that in this case, they happen to be devoid of stop codons, and that's probably why they came into existence, right? So if you were to think about what is the key event that happened to go from no existence to existence, it's actually the stochastic erasure of stop codons, which could not have been driven by natural selection. That was a stochastic process, right? So once ALTO came into being, you also actually expanded the ALTO frame, which meant that you're also expanding the large T antigen frame. And I think you asked the question, what's the impact of the large T antigen? An excellent question. So just let's get back to this idea that you've gone through these steps where really the innovation coincided. The one thing that had to happen was that the alternate frame needed to remove stop codons. And until you removed the very last codon, there could not have been any selection. That's the key, right? It's the last codon removal, last stop codon removal that allowed natural selection to say, whoa, this is a protein coding gene that we need to care about. Okay, what does ALTO do? I'm not going to give you the gory details. We don't know, but it's clearly important for the transformation phenotype. And this is, in fact, an antibody that we raised to ALTO that shows that, in fact, the protein coding gene of ALTO is expressed in Merkel cell polyomaviral infections. And this protein sequence, as you know, will have absolutely no homology to the large T antigen because it's a completely distinct string of amino acids. It's anchored to a membrane because it's actually associated with the Golgi, which is completely distinct than the large T antigen, which is in the nucleus of these cells. So you've got a completely new protein coding gene. It encodes a protein that goes to a completely distinct compartment where it actually helps the virus. So what are the implications for the large T antigen? So what I'm actually indicating here are features that are preserved in both the ALTO and the large T antigen frame, right? So on top is what the sequence looks like in the large T antigen, and in here is the alignment of the ALTO frame. Here's a case where you have preservation in the ALTO frame, but absolutely no preservation in the large T antigen. So that expansion has been facilitated not for large T antigen function, but for ALTO function. And so we've basically got this compromise now, where you've got mutations and preservation and constraint that's acting simultaneously on this piece of DNA to preserve both ALTO as well as large T antigen function. Okay, so in a nutshell, we have this something from nothing phenomenon, and that, by the way, is not rare. I would love to tell you that this is a rare phenomenon in viruses. It's very common. In fact, we don't know of a single lineage of viruses where some form of overprinting has not occurred. That's how dramatic it is, right? There's a lot of pressure to keep your genome sizes small. You want to sort of improve the coding capacity. So this is common. In polyomaviruses itself, we know of at least 4 completely distinct overprinting events arising in all different ancestral open reading frames. So it's not large T antigen related, it's other open reading frames, so in all different lineages. But what is kind of special about the Merkel cell case, which is why I presented it to you, is most of the time we don't have the transition event that tells us how did this happen. Here is a unique case where we have the ancestral state, which didn't encode for ALTO, and then the derived state, which allowed us to infer the sequence of events necessary for this completely new thing to come into being, right?
Evolution. Hello. You usually have these duplication events where you keep one copy and then you devolve the other copy. If in these kind of cases, you cannot keep another copy. You basically have to play around with this copy. So
well, it depends on how expensive it is to keep the other copy. In the human genome or a fly genome, the other copy has a long residence time before it decays. But that also means it has an opportunity to acquire a new function which is non-redundant with the original copy or a subfunctionalization. So there's ample opportunities for both copies to now become important enough to keep around. But in the virus, the residence time is slow, is not very high because there's pressure to reduce the viral genome, basically.
It's an extremely difficult problem to keep the original protein in the same reading frame and the new protein in the alternate reading frame. So you have to be it's very difficult to evolve such a gene.
I don't think that's precisely correct. I think what you're trying to say is it's not so difficult to evolve such a gene, but it increases the mutational load on the virus because you essentially, there is very little silent mutation here, right? There's no such thing as a silent mutation. You can either knock out one or the other thing. So that increases the mutational load on the virus, and that is certainly true. But that doesn't mean it's difficult to evolve the state. It's difficult to maintain the state once you evolve it. But presumably it's providing enough of an advantage that you maintain it nevertheless.
Messenger RNA, which is some long messenger RNA. So you have a long messenger RNA stretch. And now your ribosome has to latch on to it and start reading it. And it typically latches on in the correct place and reads the right frame. But any error there would cause it to produce a protein which is frame shifted, right?
Actually, it would not necessarily produce a protein that's frame shifted. It's going to go looking for a second Kosakian consensus. So it's not off by 1. It's still looking for a Kosakian consensus and a start codon. If that happens to be in the same frame, it'll basically start translating through there. So in the scanning model, it's looking for a start codon in the right consensus that's required for initiation. It's very rare that start codon will be frame shifted. It's almost impossible. There are situations where a stop codon overlaps the beginning of another thing, and that frame shifting can occur. But in a traditional scanning model, you don't get translation initiation in the -1 or the +1 frame at the original start codon.
Okay, so that's so therefore this can't happen. So it's there's very rigid.
Yeah, the original start codon cannot basically just biochemically be the source of the alternate open reading frame. It has to be at a different part of the messenger RNA. I just want to be explicit because I don't want to give you the wrong impression. So what you're asking about is you have the original start codon here, let's say, and there's a Kozakian consensus. So the ribosome arrives here. For some reason, if it cannot initiate there, it's basically going to either fall off or it's going to keep scanning till it finds something else that resembles the Kozakian consensus. But it's not going to actually misinitiate here in the wrong frame. That's distinct. Then if you have two open reading frames, this happens in retroviruses where you have ORF1, let's say, and ORF2, and you have a stop codon, but you also have an RNA secondary structure that makes it really difficult for the ribosome to scan. So what it then does, it comes here, it sort of temporarily falls off, and then it reinitiates here in the -1 frame. So, but that is actually something that is seeded because of the RNA secondary structure that is already scanning. So that's a specific feature encoded into the RNA to allow the production of the second open reading frame. So in viruses, the Kozakian consensus is very loose. So which means that if it's got a messenger RNA, it's the same original messenger RNA, and at some low frequency, the original start codon was not used, and this one was used, and now it's basically, that's the feature that's basically preserved. So we don't have any evidence that ALTO is produced of a different transcript. We think it's produced of the original transcript itself. Yeah. Yes, that's what I was describing with the ORF1, ORF2 kind of thing. It does, because actually for it to latch on to the same messenger RNA, there needs to be a very specific RNA requirement. So there is also this thing called upstream open reading frames where you have these small open reading frames. This is, by the way, true of many cancer genes. So let's say this is the primary protein coding open reading frame, but they actually have these short upstream open reading frames, and in times of stress, the ribosome will make a short protein and fall off and not produce this. But in times of non-stress, it actually bypasses this because these are not great matches to the Kozakian consensus. Go all the way here, find this, and then basically make the protein. So there's lots of ways to encode preferences for translation in the mRNA sequence. We are just barely beginning to scratch the surface of what that information content looks like. It's a really good data because there's a lot of ribosome profiling data that is coming where we know the residence times of ribosomes or different parts of the RNA. But those rules are yet to kind of come up with a clear consensus. And maybe there's no consensus. There's lots of different ways to do this. Yeah. Okay. Yes. That's right. And what's kind of cool about the system, which is why I love telling the story, is natural selection could not act on this process until the penultimate stage where it was one stop codon from coming into existence. So all of this had to have happened by neutral evolution until this frame was sort of brought into existence, and now it is basically available for natural selection to act on. So this is purely neutral evolution driven innovation, but then led to adaptability. Okay. Okay, I'm going to tell you vignette 2. I mean, all the stuff that I'm totally fine because I love the questions. So this is where we now realized that by paying more close attention to the evolutionary history of the genes, we could actually resolve a pretty raging controversy in the sort of antiviral community. Again, this is work that was done by Matt Doherty from the lab. So you guys know about this sort of arms race. We spent more than 3 hours on this. Hopefully you go to sleep and this is etched in your eyelids. But what I haven't actually told you about is, of course, this is a polynomial thing and we barely discussed it because by taking this reductionist view, I can study in arms race one gene at a time, but obviously natural selection is acting on this sort of many to one scenario. And understanding this is of course important. And we have multiple case studies where we know this is true because allelic variation provides a heterozygote advantage to the host, regardless of whether it's trying to inhibit the virus or to escape antagonism by the virus, right? So if you have allele A and allele B, you're better off against a virus no matter what, which is kind of cool. That's why diploidy is such a powerful thing. So I'm going to tell you about a gene family here called IFIT. It's not actually important that you know all of these gene families, but this just happens to be one of the most abundantly expressed gene family when the body senses that it's under pathogen attack, and MXA is also in this list. So it's called interferon-induced with tetratricopeptide repeats, because really the only thing we knew about was it's interferon-induced, and it has these TPR repeats, which are a feature that is associated with protein-protein interaction. What makes life really easy for people like me, though, is that all the IFIT genes in mammalian genomes happen to be in exactly the same synthetic genomic locus, which means no matter which mammalian genome we look at, we look to the left, we have the same genes, we look to the right, we have the same green genes, which happen to be olfactory receptors, but that's not important, which means I can look at all of IFIT evolution just by looking at this genetic location in different genomes. So there's a huge diversity you can see between human and mouse, and I'll show you more case studies where there's diversity, where there are IFIT2s, IFIT3s, and all kinds of changes. And we became interested in asking, what are the functional consequences of this IFIT evolution? But really what we were trying to do was solve a mystery. Let me just get to the mystery and then we'll get back to that. So what is the one thing that the immune system absolutely has to do in order to be successful against the pathogen? Well, it needs to kill the pathogen, and it doesn't need to kill itself. Like, very important, right? Because if it screws up on any one of those things, it's not effective. If your immune system is going to mount an autoimmune response against your own cells, that's obviously not great. And if it's not recognizing the pathogen, that's not great, which means you need mechanisms to be able to distinguish self from non-self. And a lot of the genes that are turned on in the wake of pathogen infection are actually these discriminatory genes to tell you, oh, that's a foreign gene, that's a human gene, don't touch that, go after that, and so on, right? So how do you do that? Well, one of the ways you do that is you take advantage of the fact that RNA produced by host cells is produced in the nucleus, where it's subject to this cascade of events where the RNA is capped, and then this cap is modified. So you go from a cap 0 methylation mark to a cap 1 methylation mark, where the first two nucleotides are methylated. This event has to happen in the nucleus because the machinery to put these things are only present in the nucleus, which means any virus that is replicating in the cytoplasm does not have access to these, right? And so it may go on, but it may have, let's say, a cap 0 methyltransferase, but not a cap 1. And what's really cool is, if you're a viral RNA that is modified as cap 0, you are recognized as being foreign by one of these IFIT genes, and you're not allowed to produce a protein, right? So there are lots of innovations that have occurred because of this. For instance, influenza virus will basically steal caps from host mRNAs and put it on its own mRNA to fool the genome into thinking it's a decoy. So this warfare has led to all kinds of innovation, but we're not going to talk about that today. We're going to talk about the fact that this recognition is key to the mechanism of mouse IFIT1. This was a Nature paper, it was an awesome paper, one of the first molecular cues to distinguish host from viral RNA, right? But things became quite murky because when people looked in the human IFIT1 case, they found human IFIT1 could act even though this was cap1 modified, right? So this basic mechanism, which you expect to be conserved across sort of mammalian orthologs, really was not conserved, which suggested, well, is this a mouse-specific phenomenon? So this is where we got into the picture. We said, well, let's take a closer look at the evolutionary history of the IFIT genes. So this is kind of like what the standing picture was. We had primates, which had 5 genes, and a bunch of pseudogenes, rodents that had some genes and pseudogenes, carnivores, et cetera, et cetera. And when we did a phylogeny with each of these, each of them seemed to match, except IFIT1 was like horrible. Like, so IFIT 5 from each genome seem to match. Rodents don't have an IFIT 5. IFIT 2 seem to go together. 3s go together. But these were all over the place. For instance, rodent IFIT 1s all grouped together. Primates grouped together. Dogs grouped together. Cats grouped together. This is bizarre, right? This is not what you'd expect if this was a gene family that had predated the divergence of these mammalian orders. Your first inclination when you see this is that there are many independent duplications that occurred within the same location, right? So, and that's why the cat guy is closer to the other cat guy rather than to the dog guy. You have a question? They're all on the same chromosome in the same genetic location. So this gray gene is the same in all of them. Well, we can't say that they're exactly some, of course, assembly issues with multi-gene families, but they're all in the same location. Yeah. So is there an alternate mechanism that could explain this phylogeny rather than this recurrent duplication and loss? And of course, there is, which is recombination. And that becomes evident when we do, this is ferret, by the way, IFIT 1 and IFIT 1b. What we realized was, if you look in the C-terminal third of the gene, there were all kinds of nucleotide divergence, like really divergent. But when we looked in the first 2/3, hardly any differences, right? Almost as if the evolutionary history of the first 2/3 of this IFIT gene and the last third is completely different. They might as well be completely unrelated genes, but they are not. And what this means is that, in fact, gene conversion or recombination of some sort is constantly churning out or homogenizing the first part of this gene, but not, or leaving in place the second part of the gene. And this is, by the way, true across many things. However, if you compare IFIT 1 from ferrets to panda, you see differences all throughout, which means you have gene-specific recombination that is basically fooling your and everybody else's evolutionary inferences, because after all, this is relying on the 2/3 of the gene. That's the bulk of the signal, right? So what we realized was this recombination signal is actually true everywhere, except in primates. So dog and cat and panda and ferret, they all have a recombination breakpoint independently at exactly the same codon, unbelievably. So what that means is, if you correct for this finding, and you do a phylogeny based on just the first 2/3 or just the last 1/3, you can quickly resolve this by focusing just on the last 1/3, which is actually a truer indication of the true evolutionary history of these genes. So what people had been calling 1 is actually 1b, and in fact, to make matters even worse, rodents entirely lack a functional IFIT 1, and most primates entirely lack a functional IFIT b. So when you do a blast search with IFIT 1 in human, looking for the closest guy in mouse, you find IFIT 1b, and then you complain about the fact that the properties don't match, but they are completely different genes. This is not an apples to apples comparison at all. This one has been harder to detect because of this gene conversion phenotype. However, actually any evolutionary biologist would have basically told you there's something funky going on here, and this is the merit of basically looking not just mouse and human, but across the entire mammalian lineage. So these gene families have been diverging for 100 million years of eutherian mammal evolution. But what's kind of interesting is that many species only have one or the other. And that really made it difficult to make the orthology inferences, because they're all in the same location. So when you look for orthologs, you're like, best blast hit in the same syntenic location, that's an ortholog. But in this case, that would lead to the false inference. Okay, yeah. In this group? So these guys are all completely related by orthology. So IFIT 1 is the only one that's screwy. But actually, it's a good question. We know actually nothing about the function of these guys. So functionally, we don't know anything about them, except they're all orthologs of each other. Well, not necessarily more conserved. They're just not recombining in the same way. Yeah. So this leads to the possibility that maybe both sets of laboratories were actually correct. The mouse guys had focused on IFIT 1b, and that's a CAP0 recognizer, and the human guys had focused on what ended up being IFIT 1, a paralog, and that's a CAP1 recognizer. So we wanted to test this. So here again, we wanted to do something slightly sort of unusual, which is to take advantage of budding yeast. So just like with PKR, where we took advantage of the fact that EIF2 alpha is completely conserved from yeast to human, here we took advantage of the fact that the CAP1 methylation is a property of animals. It's not found in fungi. So all yeast mRNAs only have CAP0 on them. So now what happens is if you express IFID1B here, these guys cause a growth arrest, and that's exactly what we see. But if you express the human version of the CAP1 methyltransferase, you should rescue growth, which is exactly what we see. So essentially taking advantage of the evolutionary innovation of this new methyltransferase, which is actually used to distinguish viral and host mRNAs, we can distinguish between IFID1 and 1B, because after all, IFID1 causes a growth arrest, and that is not rescued by the expression of that. So that's a completely different cue. We have no idea what IFID1 is recognizing. That's an area of hot research in Matt's lab at UC San Diego. So what about in the virus case? Well, vaccinia virus encodes this CAP1 methyltransferase. And if you delete that, you would presume that vaccinia would be susceptible to IFID1B, and that is indeed what we see. There's no difference with IFID1 because IFID1 is recognizing some other cue. However, if you do have the methyltransferase, you're immune to both of them, which means IFID1 and IFID1B, this is a long way of saying, actually are completely distinct antiviral genes that just happen to be paralogs, distant paralogs of each other, but were confused as exactly the same gene for a long time. And in fact, if you do sort of different orthologs from different species, turns out that all IFID1Bs have the property of restricting the vaccinia virus that lacks the CAP1 methyltransferase, but none of the IFID1s do. To make matters a little bit more worse, which actually really confused us for a long time, is that the human 1B, even though it doesn't have any stop codons, is basically a pseudogene. It does not have the property of encoding an intact protein. So we've got this very bizarre scenario of like a Lego block-like a distribution of IFID1 genes, some of which restrict CAP0 viruses, and others that restrict other viruses. We've actually figured out that the other viruses are like VSV-like, which are only restricted by IFID1, but not by IFID1B. And that just basically reinforces the idea that evolution is really important to pay attention to when you're doing these kinds of like cross-species comparisons. And in fact, if you want to study IFID function and you're interested in humans, mouse is pretty much the worst model system you could use. And that's actually not just true in IFID, that's true in a lot of immunity. And yet mouse is in fact the go-to model system. I apologize to the mouse people in the audience, by the way. Yeah. Of just the last 2/3, I didn't show you that. Yeah. Yeah, so the independent check is that we can look at DS, which exactly matches the DS we expect given the divergence of the species. Whereas in the first third, the DS is basically 0, which basically means it's been subject to gene conversion. Whereas the last third, DS exactly matches the cat-dog divergence or the cat-ferret divergence, etc. It's a good question. Well, monkeys are very expensive and ethically challenging, but guinea pigs actually turns out to be okay because they have the right configuration. So, but guinea pigs are really cuddly. So, I'm a fly guy, so you know, this is why we study flies. Actually, turns out now that we discovered this with IFID1, we've gone back to our own work and discovered that actually recombination is rampant when we have paralogs of the gene. So I told you that MX, which I described yesterday, is present in only 2 copies across mammals. One is usually expressed in the nucleus, one is expressed in the cytoplasm. Nonetheless, this has not prevented them from undergoing gene conversion. So in mouse, for instance, there is no MXB-like ancestral gene. Mouse has 2 MXs, but they're both MXA-like and gene converted from one another. If you basically, you can use programs such as GUARD to identify these recombination breaks. Very easy to use. Once you realize you have some sort of recombination, identifying them on the data monkey or hi-fi server is actually trivial. And so what you can see here is that if you look at the first 93 amino acids and construct a tree, it looks like this, where all the guys from the same species seem to group together. If you look at the last sort of B segment, you have exactly the expected divergence between all the MXAs and all the MXBs. So you've got like certain chunks or internal segments of the protein that are basically gene converted. Obviously, if you don't correct for this in your positive selection or any other kinds of analysis, you're going to come up with all kinds of false signals. So doing recombination as a like sanity check is really important before you embark on any kind of studies. Is that clear? Okay. This is just another example of that guard A versus guard B segments, where you basically have all the guys from the same species just grouping together, which is of course not possible in the absence of recombination. Okay, so I'm going to end with a third vignette, and if I have time, I'll maybe summarize some of the things we're doing, which is everything I've told you is assuming, again, this sort of ortholog to ortholog comparison, but a large part of our immune system in sort of eukaryotes may itself derive from unusual sources. And this is work that was actually done by C. a postdoc in our neighbor's lab at UW, and Matt Doherty. And so this premise is that, you've had host pathogen races for a long time. Is it possible that just like K3L, which is a mimic of a host defense gene, could antagonize, could you actually steal a pathogen gene, the host, and use that against the pathogen? Even better, could you actually have a bacterial warfare and having a host genome basically take advantage of that? So actually bacteria, which we care about because they kill host cells like us, but they also love to kill each other, right? Like non-kin selection. So the type 6 secretion system of bacteria is fundamentally a non-kin killing system. What that involves is contact and injection of a whole bunch of toxins from one bacteria to the other, which will basically completely kill the recipient bacteria, unless this bacteria happens to be a kin and encodes antidote proteins that prevent that killing from happening. There's all kinds, pretty much every bacterial genus has a type 6 secretion system and has elaborate forms of toxin killing. So this project started when C. approached Matt looking for a phylogenetic history coevolution between toxin genes of the deaminase variety and antitoxins of the deaminases, right? So seems totally reasonable. Let's see what the coevolution of these toxin antidote genes look like. Does everybody get what a toxin antidote gene is? Okay. By the way, that project completely failed, right? Like we had like no signal of coevolution. We could barely detect homologs of the antidotes. And what we concluded was actually the antidotes were like of completely different origin, even though the toxin were all from the same family. But in the midst of our analysis, Matt made some unusual discoveries. So remember I told you all bacterial genuses that we looked like, genera that we looked at, have these deaminases. But he also found that there were some eukaryotes that had these deaminases, right? That were clearly of bacterial origin. So here we have ticks and mites, here we have Daphnia. So we said, this is probably some sort of contamination artifact. When you're basically grinding up Daphnia, you're also grinding up all the bugs in the Daphnia, right? So you get genome sequences, which is a mix of the Daphnia and the E. coli or whatever was in it. So we decided, well, let's just tabulate this and pay attention. And what Matt realized was there were at least 6 separate instances of what should be a bona fide bacterial toxin gene that is actually present in eukaryotic genomes, right? That's just be careful like what I'm saying here, right? They're present in the genome sequences. We don't really know that they're of eukaryotic origin, but that's our starting point. So what do we do next? Well, let's pay attention to some quality checks of all, is this contamination or not? The first evidence is that these were actually part of contigs where the gene of interest, these deaminases, were in fact surrounded by bona fide eukaryotic origin genes. So that makes you feel at least a little good, right? That, okay, the genomic context in which they were discovered was eukaryotic, so that increases the probability that these are in fact eukaryotic. More impressive is they contain introns. Now, there are almost no bacteria that contain introns. So if this gene is of eukaryotic origin and it contains introns, that's actually a really, really good sign. That is in fact bona fide eukaryotic. And so all of these contain introns, which have probably been acquired after the horizontal gene transfer. So when we went back and we sort of tried to identify where all of these came from, here are the crown sort of groups of eukaryotes. And what we found was 6 separate instances. Each of these instances came from a completely different genus of bacteria. So they were not from the same. But what was cool was for the deaminase activity are completely preserved, even though they are in the eukaryotes now. So catalytically, we would suspect that they are doing exactly the same job as they were doing when they were in the bacteria. However, there's a problem. In bacteria, they use the type 6 secretion system to inject itself into recipient bacteria. How do they get out of a eukaryotic cell, right? Eukaryotes don't have type 6 secretion systems. So what was amazing was that in every instance, they have acquired a signal peptide, which basically means a methionine followed by hydrophobic of 15 to 17 residues, which is basically what you need to be recognized as something that ought to be secreted. This has happened, again, completely randomly, right? But it has happened on every instance where these deaminase genes were stolen and basically domesticated by host eukaryotes. So this is, again, basically almost smoking gun evidence that these are of eukaryotic origin, because there's absolutely no way for this kind of protein to get out of a bacterial cell. So I'm just going to present one instance. We looked at 2 of these, but one is kind of something that in the US we really care about, because ticks, of course, are carriers for many things, including the Lyme disease pathogen Borrelia. How many of you have heard about Borrelia before? Yeah, so Lyme disease is this is the causative agent. It's in fact the most prevalent vector-borne disease in the US, mostly because we don't have a lot of mosquitoes yet, but climate change is changing that. So what CMA decided to do, which is amazing, was based on our observation, she decided she's going to try to create a knockout of DAE2, which is one of these genes in ticks. The knockout CRISPR didn't work so well, but RNAi did work. You can see that she can knock down a DAE to considerable levels. Just pay attention to the log scale here. And what was kind of really interesting is when she did so, she found that the Borrelia levels basically went up by, again, a significant amount. So not only is the DAE2 preserved in all ticks and mites, this acquisition event is at least 500 million years old. It's been preserved, and it's actually doing something important to the biology of these ticks in terms of their ability to control Borrelia infection. So of course, we think about Borrelia because it's a human pathogen. Turns out it's not that friendly to the tick either, so the ticks are actually better off trying to control the Borrelia infections themselves. So here's a case of a bacterial warfare that was basically stolen by eukaryotes on 6 occasions to become a bona fide eukaryotic immunity gene. If you think about what was involved here, nothing, right? Horizontal gene transfer made a perfectly available toxin available to the eukaryotic genome. All you needed was a mechanism to get it out of the eukaryotic cell by the acquisition of the signal peptide leader sequence. But by random mutation, you can actually acquire that at very high frequency. So this is a really, I think, the tip of the iceberg of how much of our immunity, when I say our, I meant like eukaryotic immunity, may actually be of bacterial origin. After all, I mean, they're past masters at killing other bacteria. That's right. 6 independent times the transfer occurred and was preserved. There could have been many instances where it occurred but was not preserved, so we would not have been able to identify it. And each case, it came from a different bacterial genus. Yeah. We have no idea how it came from bacteria to eukaryotes, except that eukaryotes are swimming in a bacteria, right? Like you have carrying with you more bacterial than human cells right now. And I suspect that a lot of this is just coming from microbiome interactions. It could be virus involved, but I think viruses are less prone to take these phages apart. It's just much more likely that it came directly from the bacteria. Well, actually, we have very little mechanism for any kind of horizontal gene transfer. We have very good genetic smoking gun evidence that this is a horizontal gene transfer, but to catch it in the act is, of course, really difficult, right? And these events happen, 100 to 500 million years ago. So, okay. Everybody good? So I have 10 minutes or so. I'm just going to end by sort of saying, look, just in the interest of time, I focused my entire set of lectures on host-pathogen interactions. But I mean, if you think about this world of genetic conflicts, I just want to impress upon you in the last 10 minutes that there's many, many interesting things beyond host-pathogen interactions. And so this is actually relevant to perhaps other things that you guys might be interested in. And it comes back to the same kind of analysis where we can identify innovation or the potential signature of genetic conflict from these DNDS things. And even though I focused on immunity genes, I'm just going to take a few slides to focus on non-immunity genes that happen to be very rapidly evolving. I'm actually not going to show you any data because, I think, frankly, you guys have had enough from me. But I think I just want to give you a framework to guide your intuition, where if you see something in a gene category, you won't dismiss it and you'll sort of pay attention to it, okay? So across the animal kingdom, not just humans, etc., one of the most consistent categories of genes, apart from immunity and apart from olfaction, your ability to smell or taste, is actually gametic, sperm-egg interactions, right? So proteins encoded on sperm and proteins encoded on eggs are super rapidly evolving. Why is that? Okay, let's take it one by one because, I think it's worth thinking about this, right? You guys are all now masters of genetic conflicts. Let's think about it in that sort of evolutionary framework. So somebody said environment. Why would there be sort of this environmental influence or recurrent change?
It's a way of reproductive isolation.
Yeah, but why is reproductive isolation beneficial? It's actually not, right? Reproductive isolation, even Darwin realized, is actually like contrary to the theory of adaptation because you're reducing reproductive fitness rather than accelerating it. But let's hold on to that. Let's consider environment, I think, seriously, because most of the time, these, like for instance, abalone, the sperm and the eggs are literally swimming in an ocean full of like other stuff, right? So they are in fact exposed to the environment. But is the environment really consistently changing enough that it would cause this signal of recurrent rapid evolution, right? So we can distinguish between adaptation to an environmental change once versus a recurrent change to the adaptation, which is what we see here, which is what you see in immunity. You are, by the way, completely right in the sense this does lead to reproductive isolation, but that does not necessarily mean that that's the causative agent of reproductive isolation. Okay, there's somebody else who said male-female coevolution. What does that mean to you? You're on the right track, but I don't know who said it, so I don't know who. Whoever said it, you're on the right track, so maybe you want to expand on it.
Does it mean basically the sperm and the overfusing better, identifying themselves better?
So the sperm and the oocyte proteins are identifying themselves better. So let's get back to this idea of coevolution, right? So here's your adaptive space. Here's this optimal interaction between sperm and egg. And so you start here, and then you have changes in sperm, then you have changes in eggs, and you're going closer and closer. So as you get to this optimum where you recognize better, adaptation should stop, right? It should decelerate as you get closer to the optimum, but that's not what we see. We see this constant, like almost immune pathogen-like interaction. Is it? We just excluded the environment because we could do the same thing with the environment. Okay, what do we know about an average male, no matter what animal species? How many sperm does it produce relative to eggs? So the investment in individual sperm is typically 10 to the 4th or 10 to the 5th less than what you invest in an egg. Okay? The maximum intensity of selection for sperm is what? Fighting other sperm to the egg, right? That's an intense competition. The whole game for sperm is to be the one that actually gets up fertilizing the egg. That's the game. If it doesn't do so, try again in another 1,000 years, right? So the whole game for sperm is to be the one that fertilizes the egg. What is the game for eggs? Of course, they want to be fertilized. Otherwise, there's no evolutionary advantage. So are the interests exactly the same? There's one key difference, which is, I don't blame you for not knowing this, which is that for an egg, being fertilized by 2 sperm is lethal. Polyspermy is almost entirely lethal. So which means that whereas sperm are in constant competition to increase binding affinity to the egg so they can fertilize it, the egg proteins, paradoxically, are trying to impede binding affinity, not so much that they don't fertilize the egg, but to prevent the incidence of multiple sperm entering this egg in an attempt to fertilize it. Does that sound like something you've heard about before? Biochemically, that is exactly identical to what is happening in a host-pathogen interaction. One party is in the business of increasing binding affinity, and one party, even though it's a genetic collaborator, is in the business of decreasing binding affinity, because that's what actually allows it to be. So that's the reason why you don't arrive at an optimum, because what is optimal for the sperm is actually not optimum for the egg, right? So here's a feature where you ultimately expect collaboration, and there is collaboration, and yet biochemically, these two have slightly different vested interests. So this is just an example of something that should be symbiosis, but if you look at the gory biochemical details, is not. Okay. Let's consider another example. Mitochondria and nuclear proteins that work in the mitochondria. Why could this be a genetic conflict? Why could this be something that resembles a red-queen interaction? We are totally in the wild speculation part of the lectures, by the way, so there's no bad answers, but we'd like to see, how much has your intuition been served by these kinds of interactions, right? So what do we know about mitochondria, and why would they be at all in genetic conflict with nuclear genomes?
Derived from bacteria, they may appear like pathogens?
Perhaps, yes. What if I told you that this mitochondrial nuclear coevolutionary red queen is true in plants and animals, but is not true in fungi or protozoans, even though those mitochondria are all coming from the same source? So what if the red queen between mitochondria and nuclear does not play out in fungi, but does play out in both plants and animals? Your sperm and egg in plants and animals, you're already, like before, you're almost there, but I want you to expand more. That's it. Mitochondria are only passed through the mom. So for a mitochondrial DNA, it absolutely does not care about men. Sad news. No natural selection acting on the mitochondrial genome at all for male fitness in both plants and animals. In fact, there are mutations in plant mitochondria that will take a hermaphroditic plant, which could have male and female flowers, and will completely sterilize the male flowers, but will now have double the number of female flowers because that doubles the incidence of the mitochondrial transmission. So the mitochondria is exclusively a female chromosome. Can somebody think of an exclusively male chromosome? There you go. So the Y and the mitochondria are basically complete genetic competitors. What is in the best interest of the Y in terms of transmission is almost the opposite of the best interest of transmission for mitochondria. That doesn't mean that you don't need males, but for a nuclear gene on an autosome to be transmitted, you need a 50-50 ratio of males and females in the population, right? For a mitochondrial gene, you need a very small fraction of males, less than 1 in 100, presumably. So that creates a genetic conflict of interest. So mitochondria should be able to acquire, whether selfishly or otherwise, mutations that are specifically male harming. And the nuclear genome needs to actually counter-evolve in the interest of recovering male fitness to suppress that mitochondrial male homicidal behavior and try to rescue males. And that's what creates this conflict in something that is absolutely essential. So nuclear proteins that work in the mitochondria are absolutely essential. You cannot live without your mitochondria, whether you're male or female. And yet the mitochondrial genome has a say in it. So this is just an illustration, once again, of a genetic conflict in a place where you don't suspect it. And yet, if you use DNDS as your guide, it would tell you something weird is going on here, right? You could almost just follow the DNDS signature as a guide to tell you that you need to pay attention, think about the biology here, and think about it. So here, a very big clue is the fact that in fungi, mitochondria are inherited through both mating partners, but in plants and animals, they're exclusively inherited through mom. Okay, I think I will stop here and maybe take some questions, not only about today's lectures, but about everything in general. And if you guys have questions about what you're doing and how it relates to the work that I presented, I'd be happy to talk about it. We have about 10 minutes to do so. Yeah.
So we didn't say anything about, so these things will encounter different environments, different selection coefficients, etc. So somehow that never enters the picture, right? So there is some phenomenon which is common. There are a lot of parameters. So there is some parameter space, the parametric constraints such that things are common. So I'm thinking about something like evolution of sex and recombination, for example.
Yeah.
It's an unresolved problem, which is very common. So I'm just
I think maybe a simpler analogy is there are viruses that have to spend time in insect vectors and have to spend time in mammalian hosts. So you would think that, for instance, these are completely different immune pressures from completely different organisms. So why is it that an overprinting gene that would be important, for instance, in a mammalian, why would that not be lost in an insect thing? I think the way I interpret it is that the selective pressure that it encounters to reselect for the alternate frame is frequent enough that it basically is kinetically one step ahead of just a null mutation that gets rid of it. So, for instance, there are these viruses like chikungunya. This experiment has been done. Chikungunya is an insect-borne pathogen, but goes through mammals as well, right? And where it can cause illness. But in the laboratory, you can passage chikungunya just in insect cells, right? It's an easy experiment. And when you do that, you realize that some of the things that chikungunya has in its genome start decaying with no problem in terms of its infection in immune cells, so that in insect cells. That suggests that, in fact, chikungunya has two sets of repertoires for insect cell infection as well as for mammalian infection. But because in its normal life cycle it's encountering both hosts, it preserves both, because that's what it happens. But if you had to absolve it from its infection in one system, you would of course create an environment where you create relaxed selection for that preservation. So I suspect the reason these overprinted genes, which may be tissue specific, which may be only in the context, one context important, that context is encountered frequently enough that you basically preserve it. For a pathogen, it's easier to imagine why that would be the case, because viruses have no ability to live outside host cells, right? So they're constantly under that selective pressure to be able to commandeer a host cell in order to propagate. Absolutely. But I think it's simpler than that, right? I think it comes down to the fact that the fundamental, I'm looking for chalk, but I mean, as long as the virus has a certain genome size and a certain mutation rate, it basically is constrained, right? In terms of its, what is the landscape it affords? So it's even simpler than that, which is how much complexity can it encode in a genome that is naturally constrained because of sort of its mutation rate, right? So, and this is independent. This is almost like a biophysical construct. If Mike Lynch was here, you have already heard about this, that this has almost nothing to do with the lifestyle or the time scale. This is just to do with probability of lethal mutations, right? And so if you have viruses that are large genomes, they by definition have low mutation rates. Otherwise, they'd basically be, just carrying vectors for dead mutations. So if you're looking in this range, where a lot of viruses live, you have to be able to, get novelty introduced there because you're constantly fighting this battle. And I just interpret overprinting as a really clever way to introduce novelty. Now, each instance of overprinting, what you're saying is absolutely true, right? Each instance is completely idiosyncratic in terms of the context that it occurred. But the phenomenon of overprinting is almost certainly a consequence of this sort of pressure to maintain a small genome size. Does that answer your question? I don't know if I did or not. But it's a great thing to model, right? I mean, there's been almost no modeling, and it's a absolutely prevalent phenomenon. And we are getting more and more viral sequences, so more and more opportunities to be able to trace the onset of an overprinted gene. So it's a great thing for somebody who want to be studying sort of genetic novelty.
If you look at the genetic code and ask, that's supposing different triplets are juxtaposed next to each other, and then you make a shift, and therefore you will get a shift of changes of amino acids, which are going to be.
So who's likely to not give you a stop codon when you frame shift or something?
Yes, that's one kind of question. But also, how often would you expect a sequence that even folds properly, right? I mean, is there some character of the code which permits this fact that if you do a frame shift, typically, if you do a frame shift, you will get something reasonable, or that a frame shift will give you something which is completely, outside the pale?
Yeah, I can't answer that question directly because that experiment hasn't been done. But a very clever cousin of that experiment has been done by Dieter Tautz and a couple of other folks, where they literally took random strings of nucleotides with the only pressure being that they don't encode nonsense mutations, no stop codons. And there were 150 nucleotides in length, so an average protein being produced from them would be 50 amino acids long. But they're random, right? They're not of E. coli origin. They're like generated by a computer random generator. They put them into E. coli. And they were all tagged, so they could now do a selection experiment. This was a very nice paper in Nature Ecology Evolution. And they said, these sequences, which have never really even been in a cell, they've only been in a computer, if I did selection, how many of them produce something that actually impairs E. coli fitness? And that number turned out to be 20%. So whatever they were doing, a misfolding or an aggregation, etc., was enough to detectably impair E. coli fitness. But amazingly, 7% of them increase E. coli fitness, in one case by almost 100%. So something that had like absolutely no value or selection, like literally came out of a computer. So I actually think that we have grossly underestimated the ability of random sequences to give rise to. So there's this whole phenomenon of de novo genes, which is distinct from overprinted genes, but they have all the same sort of characteristics. And there are lots of de novo genes. In Drosophila, we know of at least 151 validated de novo genes in melanogaster. In humans, that number is 55 and it's growing. In budding yeast, Saccharomyces cerevisiae, which is perhaps the best annotated eukaryotic genome ever, we have just in the last 4 years added 122 open reading frames of smaller than 100 amino acids, many of which don't even exist in sensu stricto species, but they're clearly present, and in 2 cases at least have been demonstrated to cause fitness. So I actually think that our sort of obsession to think about things that are preserved, and I happen to be exactly in that category, but I've grudgingly come around to the fact that actually there's a lot of innovation to be happened just on the precipice driven by neutral evolution. You're just on the precipice of creating something quite fantastic. I don't know if that answered your question. And those characteristics are things that people like Joanna Mezel and other folks have studied. And there tends to be this disordered aspect where you have disordered domains in these de novo genes. And the longer they persist, the less disordered they get. So that's a clue that perhaps you can actually, an average sort of nucleotide string is much more likely to create a polypeptide that is of some sort of disordered character.
So, this kind of property would make sort of long jump evolution or innovation sort of more possible than incremental sort of
Yeah, I think you can get some advantage right away, but rarely. But if you have something that gives you a little bit of advantage, and that's enough for natural selection to increase your residence time before getting abraded away by mutation, now you have the property to select on it and get a much better version. I think that's what it does. It just basically protects you from the otherwise, unruly effects of mutation to just get rid of. So the question is, do you acquire enough of a selective coefficient early in the process that you basically cannot decay rapidly enough? And that's, again, a fantastic thing to model, and nothing has been done. And so much data exists now that it's really worth modeling that in a coalescent framework. Yeah. Sorry, there was another at the back. Let me.
So you talked about imprinting at the evolutionary scale level, but can it happen given the life cycle of viruses at an individual patient level? And maybe causing...
So just to clarify, I'm talking about overprinting, right? Not
overprinting,
imprinting, yeah.
And
which is another genetic conflict.
Causing resistance.
So in influenza virus, there is an overprinted gene called PAX, which is actually only in less than 10% of influenza viruses that are floating around, right? And it is a bona fide protein. We know what it does in terms of targeting the host immune system. So it's absolutely true that in the context of like a viral species that's infecting a host species, you can get innovation. Whether that's enough to get innovation in a single host, presumably yes. I mean, all of these events had to originate somewhere. But of course, catching that in the act is like a obviously very tough proposition.
Curious, in the mitochondrial genome, you have these genes which are intron.
Yes.
And so is it nature's way of sort of shutting down any slippage of the frame shifting?
Well, it's intron-less, but you have lots of frame shift that occur in plant mitochondria. You just have very few genes in the animal mitochondria. Animal mitochondria are typically 7 to 11 kilobases. In plants, they can be 50 to 200 kilobases. So lots more opportunities for innovation just by like random innovation. So I think that's primarily the reason why these male harming mitochondria have been more readily detected in plants. And by the way, this also happens to be true for chloroplast genomes, which are also only transmitted through maternal things. So you can have selfish chloroplasts just the same as you can have selfish mitochondria. Okay, last question.
Mitochondria are also under the pressure of reducing their genome size, right? So does mitochondrial genes also have overfitting?
Mitochondrial genes, we have not detected any mitochondrial gene overprinting. I just want to clarify, mitochondrial genomes are not under pressure to reduce size. That reduction in size has occurred because there's been a lot of traffic of mitochondrial genes to the nuclear genome. And then once it goes to the nucleus, there's relaxed selection to preserve that in the mitochondria. That's a Muller's ratchet. It's not because of natural selection pressure to reduce size.
So you would, so mitochondria can expand their size generally nowadays. If in the present...
Well, as I said, plants have like large mitochondria, much, much larger than animals.
Okay.